Hello everyone, I welcome you guys. Always remember, every star shines a little differently. You are a unique person, so be you. Today, topic is mechanism of innate immunity. So let's begin with the definition of immunity. Immunity is the resistance offered by the host against microorganism or any foreign substance. Immunity is of two types. Either it can be innate or acquired, which is also known as adaptive. As the name suggests, innate means this type of immunity is present right from the birth, and acquired or adaptive means it is acquired during the course of the life. Now, innate immunity is classified into species immunity, racial immunity, and individual immunity, whereas acquired immunity is broadly classified into naturally acquired and artificially acquired. So, whether we are acquiring this immunity by natural means or artificial means, that's why these two classification is there. But again, there is a subclassification called active and passive. Here, what does the word active and passive means is, active immunity means the body has to work and produce antibodies. But in passive immunity, the body don't have to work and produce antibodies. Rather, we are providing the antibodies, the preformed antibodies. For active immunity, naturally acquired, we have through infections. Whenever we are infected, our body produce antibodies against the pathogens. Hence, it is active. But take a case when the antibody is transferred from the mother to fetus. In that, the body don't have to work to produce antibodies. Hence, it is passive. Similarly, in artificially acquired, the active immunity is vaccination, in which the body has to form the antibodies against the antigen which is present in the vaccine. And artificially acquired passive immunity is from the antivenoms. We have the preformed antibodies which is directly injected. Hence, the body don't have to work for producing any antibody. That is why it's called passive. Now, coming to the classification of innate immunity, the species immunity. This is a resistance to a pathogen shown by all members of a particular species. For example, if one human is resistant to a plant pathogen, all of the humans you will see are resistant to plant pathogen. That means such immunity is present in the species. Hence, it is called species immunity. Coming to a racial immunity, different races within a particular species may exhibit resistance to infection. For example, a particular race shows a resistance to a particular pathogen, which which other races may be susceptible to it. For example, resistant to Algerian sheep, to anthrax, and other sheep are susceptible. So the Algerian sheep is is resistant to anthrax, but other sheep are susceptible to it. Coming to individual immunity, resistance to infection varies with the different individual of same race and species. For example, homozygous twins exhibit similar degree of susceptibility and resistance to TB. Such correlation is not seen in the heterozygous twins. So these are the three classification for innate immunity. Coming to the detail about innate immunity, as mentioned earlier, it is an inborn resistance against infection that an individual possesses right from the birth. Now, the most important line here is this resistance is by the virtue of genetic and constitutional makeup of the individual. Now, some peculiarities of innate immunity. It is the first line of force defense against infection. The immune response occur in minutes. Prior exposure to antigen is not required. There is no immunological memory. Diversity is limited, and it is a non-specific immunity. Factors that influence innate immunity are age, hormones, and nutrition. Now, coming to the proper topic, mechanism of innate immunity. So there are four: receptor interaction, pattern recognition receptors, and there will be components of innate immunity which we will be discussing further. Microbial surface molecules. So in receptor interaction, first of all, there will be an exposure to microorganism. Then there will be recruitment of mediators of innate immunity at the site of the infection. And as the mediators are recruited at the site of infection, there will be attachment to the microorganism, binding of the surface molecules of microorganism to the receptors on the cells of innate immunity. So in this way, the microorganism is attached to the receptors which is present on the cells of the innate immunity. Then the pattern is recognized, and we'll come over the components later. Coming to the microbial surface, these are the repeating patterns of conserved molecules. These are known as MAMPs, microbes associated molecular pattern, which are common to most microbial surfaces. Some examples are peptidoglycan, lipopolysaccharides, teichoic acid, and lipoproteins. Now, this pattern recognition receptors, PRRs, these molecules are present on the surface of host cell, example the phagocytes. And the molecules which are present on the surface of host cells, they recognize the MAMPs. For example, the host cell is the phagocytes, and they will recognize MAMPs. They are generally conserved region encoded by germline genes. Following bindings to TLRs to MAMPs, signal is generated, which cause active transcription factors. It activates the transcription factors, stimulating expression of gene encoding and cytokines and enzymes. So cytokines and enzymes will be produced, and these cytokines and enzyme will provide antimicrobial activities. The examples like toll-like receptors, these are the classical example of PRRs. These are the main receptors for inducing innate immunity. Coming to the components, the most important aspect of the video is the component of innate immunity. So how the innate immunity is provided is by anatomical barriers, physiological barriers, phagocytes, natural killer cells, complement pathways, inflammation and fever, normal resident flora, acute phase reactant protein, APRs, cytokines, and other classes of lymphocytes. Coming to the anatomical barrier, you all know what barrier means. So some barriers due to the anatomy. So we are going to provide a barrier to the pathogen due to the anatomical buildup of our body, due to the anatomy, specific anatomy. For example, the skin. So the skin mechanically prevent entry of microbes, and skin do have a sebum which has antimicrobial peptides and fatty acids. Hence, killing of the microbes by the intraepithelial lymphocytes as a typo. Coming to the mucosal barriers. So the mucous membrane entraps the microbes because it is the mucus is there. It's sticky, so the microbes get entrapped. The microbes get trapped there, and the cilia helps to propel the microbes out. And also there are normal flora there, which will helps to decrease the rate of growth of the pathogen there. So together the mucous membrane and the cilia help into the mucociliary clearance. So it will get cleared also. One thing I would like to add in anatomical barriers is the skin. In a burns patient, since the skin is lost, that's why the patient get more susceptible to infection. Hence, you can know the importance of skin in innate immunity. Coming to the physiological barriers, we have three: temperature, low pH, and secretory products of the mucosa. So the temperature, the normal temperature of the human body, this itself inhibits the growth of some microbes. Presence of low pH, for example, the gastric acidity inhibits most of the microbes. We used to read that one use of HCl is to kill the bacteria and the germs which are coming with the food. And coming to the secretory products of mucosa, secretory products of mucosa, we have saliva. In saliva, we have enzymes which damages cell wall and cell membrane. Tears. Tears contain lysozyme, which destroy the peptidoglycan layer of the cell wall. Lysozyme is the slowest enzyme, and it helps to destroy the peptidoglycan layer which is present in the bacterial cell wall. Gastric juice, as we discussed, HCl kills the microbes. Trypsin is a proteolytic enzyme. It hydrolyzes bacterial protein. Then we have bile salts, which interferes with the bacterial cell membrane and hence damages the bacteria. Fatty acids, it denatures the bacterial protein. Spermine, it is present in the semen and it inhibits gram-positive bacteria there. Lactoferrin, lactoferrin binds to iron, interferes with acquisition of iron by bacteria. So the bacteria, again in these all methods, the growth of the bacteria is checked. Coming to the phagocytes. Phagocytes rapidly recruited to the site of infection. The examples are neutrophil, macrophages, and monocytes. The process is known as phagocytosis, ingestion and the destruction of the microbes. So you can see in this diagram, the bacteria gets attached to this pseudopodia, is ingested and forms a phagosome. Then the phagosome is fused with the lysosome, form phagolysosome. Then the lysosome, due to the lytic enzymes, the bacteria get digested, and hence the digested products are released by exocytosis. This is the process, but for the recruitment, what happens is there will be chemotaxis as the bacteria and the microbes produce toxins. Then comes the attachment, ingestion, and intracellular killing. Coming to mast cells, these are activated by microbial products binding to toll-like receptors or by IgE antibody-dependent mechanisms. These present in epithelial lining of the mucosa and release cytoplasmic granule rich in histamine, cytokines, and prostaglandins. So these mast cells, which are normally present in the epithelial lining of the mucosa, these are activated by the microbial products binding to TLRs, or can be activated by IgE antibody dependent mechanism also, which we'll read later on allergies. Now, the granules, the cytoplasmic granules of mast cells are rich in histamine, cytokines, and prostaglandins. These initiate inflammation and uh, helps in killing of the bacteria. Also, these are proteolytic. Coming to natural killer cells, they kill the virus infected cells and the tumor cells. How? By releasing perforins. So perforins is like it create it perforates the cell wall, and hence the granzymes can get now enter the cell and destroy the cells. It also releases cytokines. Dendritic cells, which is an APC, antigen presenting cells, these produce cytokines to initiate inflammation. It serves as a vehicle in transporting the antigen from the skin and mucosal sites to lymph nodes, where they are presented to T cells. So as I said, it is the antigen presenting cells. They present the antigen to the T cells. Hence, they forms a bridge between innate and acquired immunity. Complement pathway, there are three: classical, alternate, and Manos. The classical pathway is antibody dependent, hence we will not talk about it in innate immunity, but the alternate and the Manos binding pathway are in the innate. What happens in alternate pathway is it is activated in response to bacterial endotoxins. So bacteria is there, they produce endotoxins. Bacteria is there and they have endotoxins. Due to that, this complement pathway is activated. Mannose binding pathway is stimulated by mannose carbohydrate residues which are present on the bacterial surface. What happens? The lysis of the target microbes by forming pores, stimulating inflammation by secreting the mediators, inflammatory mediators, and stimulate acquired immunity. Complements are also a bridge between innate and acquired immunity. The inflammation. The inflammation is defined as a biological response to a vascular tissue to harmful stimuli such as microorganism or other foreign substance, tissue injury or irritation. What happens in inflammation is there will be vasodilation, leakage, recruitment, engulfment, and destruction. So how vasodilation occurs is due to the release of the vasoactive substance from the damaged tissue, hence vasodilation is there. As there is vasodilation, so increased blood flow will be there. Due to that, due to the vasodilation also there will be leakage of the plasma protein through blood vessels. And also the phagocyte, the neutrophils and all uh, get out of the vessels and get recruited at the site of injury. And after recruitment, once the neutrophils are at the site, they will do phagocytosis. Engulfment, now this continues the steps of phagocytosis. Again, the engulfment will be the phagosome, phagolysosome, digestion, and hence digestion. So, the bacteria is there, tissue damage occurred. The mediators induce the capillary permeability. Once the permeability of the capillary increases, so the neutrophils can come out of the vessel. Then extravasation happens, phagocytes migration occurs. So chemotaxis is there. Phagocytes and exudate destroy the bacteria by phagocytosis. The process of this extravasation is known as diapedesis. This margination is when the leukocytes get attached to the vessel wall. The fever is a protective mechanism, it's a natural defense mechanism, and stimulates the production of interferons, recovery of viral infection, and destroys the pathogens, called as fever therapy. Because that we have read the normal temperature only, the bacteria are not able to, the pathogens are not able to thrive. So if there is fever increased than the body temperature, it checks the growth of the microorganism. Normal resident flora lining intestinal, respiratory and genital tract exert several antimicrobial activities, and they compete with the pathogen also. As I have told earlier, the flora, they compete with the pathogens and hence checks their growth. Also, they produce some antibacterial substances. Coming to acute phase reaction proteins. Why they are called acute phase reactant protein? Because we will be understanding by these two things, positive and negative. There are two types, positive and negatives. So acute, because in acute inflammation, if the level of these proteins increased, then those are positive APRs, and if it decreased during acute inflammation and cause a negative feedback that stimulate the liver to produce positive APRs, then it is called negative APRs. The acute phase reactant proteins like CRP, C-reactive protein, these are all diagnostic to see whether the underlying inflammation is there in the body or not. The primary site of synthesis for APRs are liver. Other sites also like endothelial cells, fibroblast, monocytes, and adipocytes. Same. Coming to cytokines. In response to microbial antigens, dendritic cells, macrophage, and other several cells secrete cytokines. They mediate cellular reaction of innate immunity. We have TNF alpha, interleukin 1, 6, 8, 12, and 16, TGF beta, and interferons alpha and beta. Lastly, coming to the other classes, the lymphocytes. Several rare types of lymphocytes that share the features of both acquired and innate immunity. We have gamma delta T cells, beta 1 cells, NK T cells, and marginal zone B cells. These are intraepithelial lymphocytes present in epithelial lining of skin and mucosa. Gamma delta T cells, beta 1 cells we have in epithelium and lymphoid organs. NK T cells we have mostly in the peritoneal cavity and the mucosal tissues. Marginal zone B cells is present in the edges of the lymphoid follicles of the spleen. So finally, these 10 components are there, and we have discussed all the mechanism of innate immunity. Thank you. We'll get back to you soon. Be you.